Greetings and welcome everyone. It's a pleasure to introduce you our first webinar of 2024 today on pests and diseases, a threat to seaweed cultivation, introduced by Professor Sylvain Faugeron. So I'm Melanie Kreff. I'm the scientific officer for the Global Seaweed Coalition. And on the behalf of the coalition, I'm delighted to welcome you all. So first, a few words about the Global Seaweed Coalition. We are a global partnership established to support the safe and sustainable development of the seaweed industry. So we have 4 action pillars: advocacy, policy, science and technology, and funding. So we fund projects through call for proposals. We inform on science and technology, like we are doing today through webinars. And we make policy change at a local and at a global level. So we've been established in 2021 as a Safe Seaweed Coalition, and we are fully funded by Lloyd Register Foundation for a program initiated for 3 years. We have the support of the CNRS, the French National Research Center, and UNGC, United Nations Global Compact, which now houses the coalition. So we have funded 1.1 million projects globally through 2 calls for proposal. And our 3rd call is actually ongoing, and we will deliver new laureates by the end of the quarter. So GSC members represent the entire seaweed value chain, from smallholders to multinational businesses, research institutes, and governmental institution. So they all work together to realize the full potential of the seaweed sector. Okay, so a couple of housekeeping detail now. So we have a chat, so please feel free to raise your question and comments. Azedine, our communication manager, and Sophia, our project manager, will be moderating it. Also, if it seems obvious, I will ask you to stay muted during Professor Sylvain Faugeron's presentation, unless called upon. Okay, so now turning into our events. So we'll give you scientific information on pests and diseases on seaweed, and especially in the case of seaweed cultivation. Professor Sylvain Faugeron, based in Chile, will introduce his work on seaweeds and give a focus on Gracilaria species, his specialty since he started his career. Now, going through the agenda. So my director and scientific director of the Global Seaweed Coalition will introduce our scientific work and the role of our scientific council within the coalition. Then Professor Sylvain Faugeron will run his presentation for about 30 minutes, and the session will be followed by a 10-minute Q&A. I will wrap up 5 minutes to the hour. But before I turn over to Philippe, we will take a group picture, so please turn on your camera so that Azedine can take the picture. Azedine, over to you.
All right. Hello, everyone. Please switch on your camera so that we have a nice screen. Good to see you all, please. Thank you. No. 3 more, please. Thank you, Maryse. All right. You can stop smiling, but keep smiling, please. It's okay. We're good to go.
Okay, thank you. So Philippe, over to you.
you, Melanie. Hello, everyone. It's really a pleasure to welcome you at this webinar, the first one of the 2024. As Melanie was mentioning, this series of webinars is part of the scientific activities the Global Seaweed Coalition is promoting through the action of its new scientific council. In 2023, we have created a fully independent scientific council, which is composed of 15 members from all over the world and with renowned scientists, which are developing activities to bring advices on also position for orientating the Global Seaweed Coalition. So among the activities this new scientific council is having, we have the intention to be present and to show important position from scientific position from the Global Seaweed Coalition through the writing of position or opinion papers. At the moment, we are working on the first opinion paper with our scientific council. It is part of the activity which will be very important during the coming year. But of course, we are also willing to share and to collect all scientific advices which could be important for the scientific for the science-based development within the Global Seaweed Coalitions. And this series of webinars we are organizing is part of it. Some of the webinars we had last year were given by some members of our scientific councils, but we are inviting also other renowned scientists to provide their view and to comment about their research, which is very important for the development of the seaweed sector. So and today it's really a pleasure to welcome Sylvain Faugeron. Sylvain is an old friend. We have shared more than 25 years of research together, and I was in fact involved in the establishment of his PhD project in Chile when he started to investigate pests and diseases in red seaweeds in Chile. And we also work together in postdoctoral projects, which involve, in fact, a large part of what he will present today in terms of how important are the problem of pests when you are managing seaweed cultivation, especially Gracilaria cultivation in Chile. And I'm sure you will be delighted by his talk. He will also, of course, develop his current research, which is conducted within a new project, a very important project in Chile to develop seaweed aquaculture, like the MASH project. He will comment probably a bit. So really, the floor is yours, Sylvain, and I wish all of you a very good seminar.
Thank you very much, Philippe, and all the global coalition. It's a very good pleasure to share some of the insights we have been accumulating from the past 25 years about seaweeds and cultivation and the problems of cultivation. So I'm sharing my screen. I hope you can see my screen. Is it okay?
Yes, we can.
I'm just trying to hide the bar in Zoom. Do you know how to do that? Okay. Pointer, sorry, I'm just finishing. Okay. So the idea is I was asked to talk a little bit about the research we're conducting related with seaweed cultivation, but mostly with the problems of epiphytes. I mean, I will talk a little bit about this. I'm in a Chilean university, but I am also part of a joint laboratory with the CNRS that is called Evolutionary Biology and Ecology of Algae. And more recently, we have built a group that Philippe was mentioning that is called the Marine Agronomy of Seaweed Holobionts, and it's trying to gather scientists and producers and try to develop this concept of marine agronomy that so many people in the world are trying to promote and in a way transfer science to producers and help improve productivity. So why Chile? Well, Chile is actually one of the big actors in seaweed production. It's the sixth country producing and exporting biomass of algae, and this is quite relevant. You might say it's far behind other Asian countries, of course, but it's the first non-Asian country that is producing quite a lot of biomass. But when we say producing, actually, it's mostly harvesting. And as you can see here, it's 90% or more of this biomass actually comes from natural population, and less than 10% of the biomass comes from cultivation in farms. And this is a real challenge here in Chile. We are, since many years, trying to boost the aquaculture of seaweeds. And it's a slow growth. It's a slow progress so far, but at the same time, Chile has a history of around 45 years of seaweed farming. So it's not a new activity. In Chile, the main algae by far, it's almost the only one, or most of the biomass comes from what we call here the pelillo. Pelillo is a red algae, Gracilaria chilensis, that is cultivated, as you can see, in estuaries and in sandy beaches. At low tides, there are many places where the tide is sufficiently high so that the people can reach large pieces of intertidal area. And it's about a little bit more than 5,000 tons per year that are being produced through this production. There are about 600 farms, a little bit more. It's fluctuating yearly, but the last statistics estimate about 600 farms producing pelillo in Chile. They are producing essentially in the central south part of the country, so that's where Patagonia begins. It's a place where there are many islands and fjords and sounds, and it's especially adequate place to cultivate Gracilaria because there are not too many waves. The water is calm, and there are essentially tide currents. And the cultivation it's mostly artisanal. There are a few companies, but usually it's more a family matter, and so it's extensive cultivation, small pieces of the coast that have been colonized by farms, and it's usually a complementary activity of these families. So quite about 25 years ago, we started having a look about the effects of this cultivation and what were the main changes that we could observe between natural populations and cultivated populations. At that time, we were interested in different aspects, essentially related with the domestication process. And we have been measuring traits that could mark some difference between natural and cultivated populations, and that we could eventually interpret as initial signatures of domestication, or incipient stages of domestication, if you want. So, for example, when you go to farms, each bar represents a farm here. Most of the farms are dominated by diploid material. So Gracilaria has these typical haploid-diploid life cycles, and it's isomorphic. You have exactly the same morphology of the males and the females that are haploid, and the sporophytes that are diploid. Nevertheless, in the farms, we find mostly diploids. And it was quite surprising because the cultivators actually do not know how to recognize haploids from diploids. They just select the fastest growers during their propagation. The biomass is essentially propagated by clonal fragmentation. so they select the ones that grow the fastest. And by doing so, they actually selected the diploids that are dominating in the farms compared with natural population, in which we find about half of the population that are diploids and half of the population that are haploids. Associated with these type of traits, we also notice that in farms, usually the individuals are vegetative. They do not produce reproductive structures. Whereas in natural population, as you can see, almost 100% of the individuals produce reproductive structures. Of course, natural populations occur usually in rock pools. So in order to colonize rock pools, they need to produce spores that settle on the rock and then produce the new individual. So this is how the natural cycle is maintained. In farms, because of this clonal propagation, then sexual reproduction is no longer needed. In fact, there are some hypotheses that we have not completely clarified yet that suggest that the selection for the fast growth probably goes at the expense of the production of reproductive structures. But also, there are some hypotheses related with the clonal reproduction itself that allows sometimes the accumulation of mutations, and some of the mutations might affect traits that are not needed in farms. So, for example, the production of reproductive structures. So these suggest that there are changes in some traits, and these changes can be the result of the selection by the farmers and probably represent a signature of incipient domestication. And among the traits that change and often change quite a lot between natural population and farm is the epiphytic load. So on the left side, you can see a typical Gracilaria individual growing on a rock pool. So this is how it looks like Gracilaria, and as you can see, it's relatively clean thallus, free of epiphyte, at least macroscopic epiphyte. And on the right, so you can see something that is a mixture of different seaweeds, and actually it's a thallus that I collected from a farm. So this line, this darker line here, actually is the Gracilaria, and all the others are epiphytes growing on Gracilaria. So this is how sometimes it looks like a cultivated Gracilaria in Chile. And this is quite an extreme case. It's not always like that, but there is quite a lot of epiphyte pressure on this tally in the farms. So we had the different questions that we have been addressing to try to understand why is this specific epiphytic load in farms. Is it the consequence of domestication? For example, is it the selection for fast growth thallus that actually have an indirect effect on the resistance or susceptibility to epiphytes? Is it just the fact that there is a dense population of clonal organisms, so they are relatively similar genetically, and this is usually a very good context for pests and pathogens to grow, because once they infect one individual, they can actually infect all the others because they are exactly the same and they are very close to each other, so it's easy to expand in this type of populations. So what I'm going to talk is a little bit of how we have been approaching this problem. Unfortunately, we don't have any solution for the epiphytism yet, but the idea is to show you that we have been trying to address several questions and trying to explore some possible routes towards eventually some solution. So the first thing we have been doing was to characterize this diversity of epiphytes. So epiphytism usually concerns a lot of different species. And we noticed that these different species not always cause the same type of impact on the host. So we created a kind of classification of the type of damage that is being caused on the host from type 1, on which the epiphyte is just settling on the surface of the host. So here it's the host, and here is the tissue in electron microscopy. So it's a transversal cut of the epiphyte, and this is the interface, the contact between the two. And what we can see in type 1, so that the epiphyte is present, but the seaweed, the host seaweed, is not noticing there is no visible change in the morphology of the algae. In type 2, there are very minor changes in the morphology of the algae, but what is really present in between is the bacterial communities that are really growing in a very dense and very thick layer. So we see, in opposite to the type 1, that in type 2, there are essentially a bacterial biofilm that is growing in between the epiphyte and the host. And they, type 3, type 4, type 5, are showing different types of impacts. So, for example, the cortical layer can be disrupted, or eventually there can be some kind of abnormal tissue growth that could represent a healing process, but also some kind of abnormal growth, like in a cancer, in a tumor, that cells start to grow in a different ways as they are supposed to be behaving. So this is the epiphyte here, and this is the contact, and you can see that the cortical cells are no longer typical cortical cells. They are developing a kind of gall at the contact zone. And in type 5, there is this penetration of the epiphyte within the host tissue, and it's an epiendophyte in this case. And there is, of course, associated with that a very strong damage to the host tissue, eventually infection by many bacteria and fungi, and then the degradation of the host. When we try to analyze the dynamics of these different epiphytes, so we can monitor through the year, among different years, and in different farms, and we can see that in fact there are a lot of diversity in these dynamics. In some cases, different epiphytes behave relatively synchronously, so that means that they all grow at the same time, and it can reflect seasonal variation, and probably associated with the productivity of the environment. But sometimes, like in this case, we can see that it's not all the types of epiphyte that are growing at the same time. Some are present throughout the year, some others have a typical seasonal pattern or appear for very few months. And this imposes specific challenges, because if the farmer wants to control the epiphytes or schedule eventually the harvest before the epiphytic bloom, so it has to be very specific to the type of farms and the type of place the farm is installed, because these dynamics are highly variable. And eventually he will focus on specific epiphytes as well. The farmer can decide to avoid the growth of specific epiphytes, the ones that cause most damage or the ones that cause most cover, because the more the cover of the epiphyte, eventually the less the quality of the algae, and so the lesser the price that he can sell. So analyzing these dynamics have proven to be quite important for the farmers themselves. And this is just one of the examples that we have been analyzing. By doing this type of monitoring, we noticed that some strains eventually were always less infected than others. And we have been isolating these strains and developed some bioassays to analyze whether this was a consequence of where they were grown or whether it was something associated to a resistance to epiphytism. And as you can see, so these are different strains, and the ones that we call resistant versus susceptible, it's when we notice, for example, that the epiphyte settles easily or less easily on the surface of the host. So this In this case, it's a bioassay done with a red algae used as epiphyte. It's an acrocatium. It's a small filamentous algae. And in the resistant strains, actually, we notice that there are less spores that settle on the surface than on the susceptible ones. And susceptibility and resistance tends to be relatively universal. So it means that once it's efficient against one epiphyte species, it can be efficient towards other epiphyte species. In this case, it's the same bioassay, but done with another red algae, Erythrocladia, that form an encrusting algae on the surface of the host. It's a type 3 epiphyte, so it's causing a damage on the host, as we can see with the depigmentation of the host cells. And so on the susceptible strains, the spore settles and germinate and develop the crust on the host, while on the resistant strains, the spores settle less, but even the spores that settled, that managed to reach the surface of the host, actually, they remain as a small spore or with very few cells, are germinating with very few cells. They do not form the typical crust. So apparently there is something that some strains are able to do to repel the arrival of the epiphyte or to control their development on their surface. So one of the questions was whether the typical epiphyte load observed in farm was the consequence of actually selecting strains that would be more susceptible than if compared with natural populations, for example. Do we observe more resistance in natural population than in farm? That was more or less the question that we wanted to address. And we started to compare strains from different natural populations and strains from different farms, the males and females haploids and the tetrasporophytes, the diploids. Remember that only diploids are present in farms, so this is why there is only this category here. And we tested with different types of bioassays with different epiphytes, just to explore the phenomenon of the variability in the response to epiphytes. And what we have shown, for example, is that usually there are no major differences between farms and natural populations in terms of response to epiphytes. This is done with acrocatium as a bioassay, and actually, when we compare with a glass slide, on which we settle the spores, it appears that these algae settles and grow much better on the Gracilaria than on a glass slide. So apparently, Gracilaria is helping this epiphyte to grow compared to an inert support, but there are no major differences between farmed and natural populations. We did the same with Ectocarpus, that is another type of epiphyte that we find regularly on Gracilaria, and actually it's the same phenomenon. We don't really see differences between farms and natural populations. So apparently it's not really, the high epiphytic load in farm is not really related with a specific selection of this type of susceptible strains. But to understand this mechanism, also the resistance, the apparent resistance of some strains to epiphytism, then we went to explore possible mechanism of response to the presence of epiphytes. So here on the top left, we can see that when we inoculate the Gracilaria with acrocatium or with Ectocarpus, and we compare natural and cultivated individuals, we see that there are some changes associated with the total phenolic content. So the phenolic compounds are usually considered as compounds that help into defense against grazers or against pathogens. And we can see that when we compare with the control, the two types of epiphytes actually induce a change in the total phenolic content. So the seaweed is really perceiving the species, the different species that are settling on its surface. And it's reacting through the production of phenolic compounds. And these are the differences between control and the different types of epiphyte, but the differences between natural and farmed individuals, as you can see, there are no differences. It's exactly the same for Acrocatium, and it's roughly the same if we take into consideration the variance here, it's the same. So there are no differences between farmed and natural population, but yes, there is a response to the presence of epiphytes. So part of the work has been focused on trying to identify what would be the molecules that are actually part of this response and inducing the resistance. So some of the molecules that were explored, tested are molecules that derive for the oxidation of some lipids like arachidonic acid. So this is arachidonic acid is a typical lipid that is used in plants as a defense signaling mechanism and an activator of defense mechanisms against pathogens. And these two compounds, 8-ET and D-ET, are derivative of arachidonic acids, so different compounds, but because they were oxidized. And when these compounds are tested on inert supports or on compare with Gracilaria extracts, we can see that they have an effect on the number of spores that are able to settle, to germinate, and to develop as an epiphyte. So these experiments show that depending on the dose and depending on the compound, we can have an effect that is similar to the typical defense that we observe on the surface of the resistant strains, suggesting that there has something to do with the signaling or the activation of defense mechanisms. Well, we have been doing many different experiments to try to get a little bit closer to the mechanism of defense themselves. And one of the approach, the most recent one at least, is trying to identify the genes that might be activating during the defense of the resistant strains. So we compare the transcriptomic profile, so the genes that are expressed in the certain condition in the strains that we call resistant and susceptible in the presence of the epiphytes. And we identified around 200 genes that are upregulated, so that means that are overexpressed in the resistant strain specifically when they are exposed to the epiphytes, while the susceptible strains overexpress only 40 genes. Different genes. So these are all differentially expressed genes. And when we look at the functions, so most of these genes actually have no known functions in the genome of Gracilaria. We can identify the genes, but they are mostly new genes, so we don't really know their function. But for the ones we know the function, in the resistance, among these 200 genes, we can find genes related with the response to stress. We can also detect some genes related with signal transduction, so suggesting that there are some signals that are activating responses into the host, and some processes related with the degradation of proteins that might be associated with this defense. And interestingly, there are also some responses that can be detected in the epiphyte. For example, when we analyze the genes that are differentially expressed in the epiphyte, when we put this epiphyte on a resistance host or on a susceptible host. And as you can see, there are genes that are differentially expressed, suggesting that whatever the host is producing is probably releasing some compounds that affect the activities, the gene expression of the epiphyte. And so the mechanism of resistance is probably a mechanism of defense that activates something that the epiphyte is perceiving and then eventually allowing its growth on the host or stopping or reducing its growth on the host. So this is how we have been exploring possible mechanisms of response to epiphytes. And more recently, we have been exploring another line of hypotheses or trying to address different questions related to this interaction. When we look at this interface between epiphytes and host, as I mentioned, sometimes there are quite relatively thick layers of bacteria. And we started to wonder whether these bacteria could be actually regulating the interaction with epiphytes. These biofilms eventually could have a role in this interaction. So we wanted to test this. And we wanted to test this because when we consider the seaweed, every seaweed has a biofilm on its surface. And the natural community of bacteria that live on a natural seaweed, so a natural seaweed living in a natural population, is probably something that co-evolved, meaning that the composition of the bacterial community is selected through probably millions of years of coexistence with Gracilaria in natural populations. And when the farmers take these natural strains and take them to their farm, where the environment is different, when the density of the population is different, when the selection of the fast growers is imposing some changes in the types of individuals that are being grown in the farms. So probably this also affects the composition of the microbial community. And so we wanted to see whether the epiphytism could be actually the consequences of changing the microbial community because of the farming practices. So we have been exploring these ideas through different ways, and I'm just going to show you one type of experiment that we are conducting here that are bioassays in the lab. So we develop bioassays in the lab because we can really control the genetic identity of the strains that we have been testing. We can really control the presence, absence of the bacteria. We can eventually build communities of different bacterial species or strains and test these effects of different composition of bacteria or the presence, absence. So how do we do that? First, the first step is really to get difference of the algae with its natural biofilm. So this is a scanning electron microscopy, and we can see that on the surface there's these granulous structures that are the extracellular matrix. It's the biofilm. So it's the biofilm is a matrix of the bacterial cells plus a matrix that is in which they are embedded and they grow on the surface of the host. And when we use cleaning, ultrasounds, antibiotics, and so on, we get the surface of the host completely free of these biofilms. So we can have this experimental system in which we can compare the presence or absence of the bacteria. And when we say absence of bacteria, it's really absence of the biofilm, because sometimes we can see here, for example, there is a one spore or maybe two small dots that could be some bacteria. We have seen that they are not completely absent, but the biofilm is removed. We can do the same for the epiphyte, but the epiphyte is used here as a bioassay. So we control the reproduction of the epiphyte to make sure how many spores are inoculated on the host, so that we can really quantify in a precise manner the amount of spores that settle, the amount of spores that germinate, and how many growth rates or ramification or whatever the indicator we can measure then on the epiphyte growing on the host. So this is how we inoculate, and we then count the spores on the surface. So we use because Gracilaria is a cylindric thallus, so we can look at the microscope and observe on the border from remember that on the microscope we're observing from the upper part of the thallus, and we just observe the border. So it's like a transect, and we count the spores that are settled on this surface, on this transect along the surface. We can also eventually use extracts of the algae, for example, the extract of the complete holobiont, so that's the algae plus its biofilm, or extracts of the bacteria only, or extracts of the exudates from the algae, just to see if we can. So the hypothesis is that eventually in this interaction, there are signaling molecules that might influence the development of the epiphyte. So we can test in vitro the effect of these compounds on the settlement, germination, and growth of the epiphyte without the host, just to check whether we could identify some compounds that could be regulating this. And then another type of experiment is just trying to eventually exchange the microbial community, so we can reinoculate bacteria from, for example, if we're comparing natural and cultivated strains of Gracilaria, we can reinoculate bacteria from farms or bacteria from natural populations and see whether the composition of these different bacterial community actually influence the result of the interaction with the epiphyte. So a few results of that. For example, when we compare the resistant and the susceptible strains without the biofilm, we can see that susceptible strains are always heavily infected, so the number of settled spores is very high. In the resistant strains, as expected with its biofilm, it has much less spores that are able to settle on this surface. But when we remove the bacteria, actually the resistance is removed as well. And the resistance phenomenon nearly disappears when we remove the bacteria from the surface of the host. So apparently the bacteria are playing a role in the mechanism of resistance of these strains. When we try to identify different extracts from the host, from the bacteria, from the exudates, and try to see whether these have effects on the capacity of the epiphyte to grow. Actually, so far we didn't find any interesting results. It's just, this is just to show you that we don't see major differences between the different types of extracts. The only difference we can see is when we remove the biofilm. And so actually, when there are no bacteria, the epiphyte is not really able to grow. It requires some bacterial signaling. So the epiphyte will be able to produce filaments, ramification, and the total biomass when there is bacteria in the system. If we just inoculate the exudates, so without just the molecules, without the bacteria, then there is no effect. It's just like when we remove all the bacteria from the system. So apparently there is a certain amount of signaling molecules that are present that are related to the presence of bacteria. And so far we were not able to identify these signaling molecules, but there is potentially a role of these that are affecting the capacity of the epiphyte to grow. This is another type of result when we compare the strains without biofilm compared with the ones that have the biofilms from the laboratory conditions. So it's a native laboratory biofilm, if you want. And then we compare with these other two treatments when we reinoculate from natural populations or when we reinoculate bacteria that come from farms. Here, the idea was to see whether the identity of the bacterial community would play a role in this interaction with the epiphyte. So what we see is that actually when we remove the bacteria, there is much more epiphytes that are able to settle on the surface. So again, this, as we saw before, the resistance is jammed when we remove the bacteria. There is no resistance possible. But the identity of the type of community actually has no effect. So it's really a matter of presence, absence of bacteria, but the identity of this bacteria apparently doesn't have any effect on the response of the host to this to the presence of epiphytes. We also try to test some hypotheses related with this idea that the bacterial community could have co-evolved with the host, so there could be some kind of symbiotic combinations. And on the opposite, if in the farms there is a disruption of the native bacterial communities, and it could cause what we call the dysbiosis. So it's the contrary of symbiosis. It's a presence of bacteria that will have negative effects on the host because of this disruption of the native composition of the community. And in both cases, we don't see any difference. There is no statistical difference. So clearly there is no effect of the type of microbial community and the composition of the different bacteria that are in this type of communities. So far what we have been seeing is that the presence here versus the absence there of microbial biofilm is strongly regulating the presence absence of epiphytes. These are the epiphytes. This is a spore of Acrocatium, and the first cell after the germination, so the first erected cell. So we can see that there are different spores that settle here, and they're all germinated on the surface or when there is no bacteria, and it's very hard to see these on the surface of host when there is this type of microbial biofilm. So one hypothesis could be simply that it's the physical layer of the biofilm that is somehow impeding the settlement of the spores, or there are some signals, molecules that are present and regulating this interaction, but we were not able to identify them yet. Clearly, the composition of the microbial community seems to have no effect on this interaction. And the natural and cultivated population seem to have the same pattern or response, although we usually observe a much higher variance in the response in the natural population. And this variance, this increased variance, or the differences among different individuals within natural population, suggest that there are some genetic determinants behind this interaction, because again, in natural population, we have sexual reproduction, so more genetic diversity compared with farms where there is clonal propagation and all the individuals are genetically similar. So if they are genetically similar, they can have similar response to the epiphytes. So we are still exploring this type of questions to understand and further test this hypothesis of a regulation or combination between the genetic diversity of the host and the presence absence of the bacterial communities. So to do that, there are, just to finish, some experiments. I'm just showing experimental designs because they are ongoing. Some of them include understanding the inheritance of the bacterial community and whether there is a possibility of selection of some holobiont characteristic that would be more susceptible, more resistant. So holobiont means a specific host genotype with a specific microbial community. And this supposes that there is some kind of inheritance of the microbial communities in the farms. So we are testing this type of hypothesis. And then we are testing more broader hypotheses just related with the cultivation practices. That means that when we have a dense and genetically homogeneous population, we have a facilitator for the spread of the disease. I think I will finish with, I think I've spoken a lot. I just want to thank all the collaborators. As you can see, there are many collaborators. It's more than 25 years of research. And there are probably much more than that I didn't include. And currently, so most of this research is done within this group of that trying to foster the marine agronomy for seaweeds. So thank you for your attention.
Thank you so much, Sylvain, for this presentation. Now we will open the floor to questions. I'm sure there is a few. So please raise your hand if you have any. Yes, I see a first hand for Valeska. Please go ahead.
Yes, thank you, Sylvain. I have a question related to the presence of epiphytes. Is there a relation to the salinity of the water?
Not always, no. So Gracilaria is cultivated in quite a large range of salinity because it can be in estuaries or in places where it's a normal salinity, if you wish, and apparently it's not related with salinity because we can find epiphytic spread also in the estuaries. But it's difficult to distinguish because actually sometimes the cultivation practice are different in estuaries compared to other places because there is much more current, and so they use a different system. They tend to use more ropes instead of burying the talus in the sand. So because it's also different techniques, it's difficult to test. In the lab, we didn't test salinity anyway so far.
Okay, thank you.
We have questions in the Q&A too. And we have a question from Thierry and from Flower.
Maybe raise the question from the chat, Azedine.
Okay, all right. So we have two questions. In fact, the first one is there seaweed cultivation on this farm all year round? So that's the first question. And the second question from Maris was, I will try to pronounce it well in the English way, how to obtain resistant strains just by selection? Could the selected strains be still considered as maintaining genetic diversity of local strains?
Okay, so thank you for this question. So yes, cultivation is occurring all around the year, and in some places they do 2, even sometimes 3 harvests a year. It depends on how long how the talus when they want to harvest. So it's operating all around the year. So related with selection of these strains, so of course there is a big interest in using resistant strains, and some of these have been tested in farms, and they actually remain relatively free of epiphytes during the whole season of growth. It has not really been tested in like a typical agronom engineer would do in land plants. I mean, he would be testing this in different farms and measure the performance in terms of growth and other traits. This has been done in only one farm in which we confirm that the seaweed is able to grow and able to keep clean. So what would be the consequences of settling farms with this type of strains? Yes, you're right. It's probably going to cause a further depletion of the genetic diversity because we will have one or very few selected strains. So the idea now is really to work on identifying these genes responsible for the resistance in order to make some crosses and make some integration of these genes into natural strains so that we can have a large genetic diversity on most parts of the genomes, except on these regions, on the genomic regions related with resistance. That's the plan now.
Okay, thank you, Sylvain. So we'll take the question of Thierry. I don't know who was first.
Yes. Okay, bonjour Sylvain. It's a long time I haven't seen you.
Yes.
So I work with a very different system. It's not Gracilaria, it's with kelp. It's both Saccharina and Alaria. And for us, what has been very important to avoid epiphyte is the time we transfer the spools, the ropes, when we transfer them at sea. Because I could put my kelp at sea from the laboratory to the sea, I would say between August and November. And at the end, in the next May, I don't see too much difference in length and biomass. So that but what we do is we transfer our kelp in November, late October, November, in the northern hemisphere, and that's to beat the. For me, it's a question of putting the ropes. It's putting substrates in the water. And it's a question of colonization of substrate. And early on, the kelps are very small. So if I put them too early in August, I will have the ropes covered with vulva and all these things. If I wait, I don't get them covered. The kelp have a good start in November, and I get pretty much the same biomass in May. So for me, I don't know if the chemistry is associated with that and there is seasonal change and everything, but for me, it has been a competition for substrate.
So here we have been trying to follow some of the practices of different farmers, and we noticed that the way the farmer is operating its farm is very important as well, but not necessarily in the timing of the settling. Here, what we have been observing is, for example, when the people burrow the part of the talus in the sediment, in the sand or in the mud, so often you have a rapid growth, and this rapid growth is a kind of escaping the development of the epiphytes. The epiphytes manage to reach the host, but they do not produce a large amount of biomass because Gracilaria is the one that is growing fast. And so if they harvest relatively soon in the season, then they have a clean material. If they let this in place, then at some point it just became invaded completely. The ones that work on ropes have relatively same problems. For example, when they use ropes, so they disentangle the rope, put the part of the talus in between, and then have different mesh of Gracilaria every 30 centimeters. So this is a lot of work, if you want. So it's a lot of hours putting this algae. So some of the cultivators, what they do is that they put a rope. It's a clean rope with clean algae in the sea, and they cut when they have the biomass they want to collect. They cut the algae, but they leave the rope and the small parts of the talus. And this is sometimes disastrous in terms of, because the rope and the bases of the talus are colonized by epiphytes, and then when they grow, it's all invaded. The ones that actually remove the ropes to harvest on land and clean the rope and put new material tend to have relatively cleaner epiphyte-free system. And it's also the same from the ones that now are trying to settle spores on the ropes. So they settle spores on the ropes, they have really new material, and they tend to have cleaner material, but they have a lot of variability in terms of production, so they're not really happy with that. And if they cut, then the material that will regrow from that will be again epiphytized. So it's a lot of the practices that are influencing, yes, the amount of epiphytes.
Sylvain, can you remain a couple of more minutes to take two questions? Yeah,
of course.
So there is one question from Flora, and then there is a question in the chat from Harrison, and then we will close. So go ahead.
All right. Thank you very much for your presentation. Looking at your work, I see that you have been studying these diseases for quite some time. So my question is just to ask if you have seen any difference, any increase in disease incidences and so on, caused by, for example, change of temperature, change of the environment, or climate change. Have you seen some kind of increasing incidences of diseases, or is it the same as before?
Okay, it's a very interesting question, this, because yes, there are complex interaction with climate change as in most places of the world. So in Chile, in the places, in the region where Gracilaria is mostly cultivated, is this region near the Patagonia. So the Patagonia is relatively cold climate, rainy, and the region is really influenced by rivers. And so the climate change is reducing the amount of rain. It's reducing some winds in summer in particular. And this creates, this increases the stratification of the seawater in the fjords. So the upper layer tends to be warmer and eventually with lower salinity. And this has obviously some effects on the production and the invasion of some of the pests. And what have been what some colleagues have been observing is that in the estuaries, because there is less flow of the river, the water is actually more concentrated in different compounds, including nitrate and phosphate coming from land. And this promotes the growth of many different other species of algae, especially the green ones. So the farms in some places are really invaded by especially green algae, in fact. But in other places, so Gracilaria is also cultivated in more northern parts of the Chile, so that's the desert of Atacama. And in this place, in the desert of Atacama, the global change is causing strengthening of the winds that promote upwelling of deep water. This upwelling, so it's water from the deep ocean that come to the coast that is very cold and also rich in nutrients, but on minerals, not on nitrate and phosphate. And in this case, there are specific seaweeds that are actually growing in these very good conditions. And Gracilaria is one of them, so they grow very fast, but some of the epiphytes also. So then there are some specific epiphytes that are eventually becoming more problematic in the northern part of the country. So it's really dependent on the region. And there are some tendency that are, as you said, related with the climate change. So it's not really well quantified for the moment, so I cannot have a more specific response because we don't have really hard data, but it's something to be quantified for sure.
Thank you.
Azedine, can you read the last question in the chat, please?
Yes, of course. So this is the question. Given your research, what are the prospects for resolutions of the challenges you are presenting? What kind of support would be required to expedite solutions? It's a nice question.
So that's a very tricky question. So as a basic researcher, for me, it has been really difficult to translate what we have been observing in the lab into actually not even a solution, but not even a clue of a solution for the moment. I mean, we were really expecting that the identification of some resistant strain would be something of interest, and we have been proposing this to the farmers, but so far, not many farmers have, not a single one actually, farmers have been interested in testing this in real condition. Remember that the farmers here are not really, they are mostly from fishermen families, and they're not really concerned about the quality of the production. They just want biomass to be able to sell. It's really a simple activity, not really technified, and there is no training in seaweed farming in Chile and so on. So maybe this is one reason that we have not been able to get the sufficient contacts with the farmers to test this. The other thing is so there are institutions in Chile related with the support of aquaculture, and this institution, yes, they are starting relatively recently to take into consideration the fact that eventually what is being cultivated is not one single thing. It's that pelillo, the algae, is considered as a single entity, but actually there is genetic diversity, there are different populations that behave differently, and so on. And there are these resistant strains. So they are in a process of creating a seed bank, a biobank of different strains, and we are contributing with that endeavor of these institutions. And the idea is to start to develop field tests of the different combination of genotypes in different types of farms and so on. So this is it's a recent progress, and we will see some results, I guess, in 2 or 3 years, hopefully. So interesting results, but so far solutions no more than that. And the last thing is that we were expecting that eventually the use of specific bacterial communities would help, because it has been seen in land plants that eventually modifying the microbiome can have very strong and specific effects on the development of the plant, on the resistance to diseases, on the production of fruits or whatever. And there is a lot of expectation of using microbial communities to impact in seaweed aquaculture. But so far, testing this experimentally is not so easy, and we have not found some strong evidence showing that this could be a solution.
Thank you so much, Professor, for answering all the questions and for taking the time to introduce all about your work today. Thank you all also for attending this event. So I will just make a promotional word for our upcoming webinars. So in March, we will have Flora M. Suida and Professor John Bolton introducing the seaweed cultivation in Africa. Where are the opportunities? So that will be 22nd of March. And then following that, on the 17th of May, Professor Elisabeth Cotier-Cook will introduce seaweed and biosecurity topic. Also, if you are not registered yet as a member, I encourage you to do so. It's free to access and the coalition, so you will have access to our seaweed community and to our private events. So I won't be longer than that. Thank you all for attending, and looking forward to meeting you at the next session. Have a good day.
Thank you, everyone. Goodbye.
Bye. Thank you.